In the last 50 years, there has been an increase in food demand because of the growing population. Ruminants, because of their unique digestive system, they can convert fibrous plant material that humans cannot consume in products that the animal can consume, such as meat and milk. The ruminants have a unique digestive system formed by 4 chambers, and the rumen is the biggest one and also the most important one because it's where most of the microbial fermentation of the feedstuff happen.
My lab specifically works on microbes that are found in many different environments, but specifically we are interested in microbes that live in the gut of cattle. Methane is a highly potent greenhouse gas, which contributes to a lot of the climate change that we are seeing. It's not the cow itself that actually generates that methane, it's actually the microbes that are living in the digestive tract of that cow. That means that these microbes will generate that methane that is being released, then contributes to climate change.
Most of the methane mitigation strategies are based on the addition of feed additives into the diet. In this case, what we are testing is the addition of cultures itself. So like instead of adding a compound that can alter the microbial population, we are directly altering it by enriching a microbial group in the rumen. Because we believe that the function of this microbial group in the rumen will have an indirect effect on the activity of the rumen methanogens. So at the end, we will have a decrease in methane production. We're going to collect samples from each of the cultures to extract DNA from them. So later, we will be able to determine the concentration of the cells. And the rest of the culture will be transferred to these containers. So we will centrifuge all the culture to take the pellet, and in that pellet, it will be our cells. What we do is we test these compounds in our bioreactor systems. So they mimic the same conditions that in the rumen. So we assume that the effects that we see in these bioreactor systems, we will be able to potentially see in the animal itself.
So during the breakdown of the feed, the microbes will be producing gas, and gas is going to be building up inside these vessels. These ANCOM systems, they can detect the gas pressure inside, and when it reaches a certain pressure hold, it will automatically open right here, and the gas will be released up into this pipe, and the gas will be collected in our gas bags like this. So you see, our gas bags are filled up, and will be ready for our gas chromatography to detect what gases are in there.
It's very important to test these compounds first in vitro. And then once we select the best candidate that can potentially decrease methane production without affecting the fermentation of the feedstuff, or even if that they can improve the fermentation of the rumen efficiency, then we can go to the field, we can go to the farm, and we can test this compound in vivo, in the animal.
If that energy is not going directly into milk, for example, or protein, but rather is converted into methane, it means we are losing that energy and we are losing valuable feed that is additional cost to the farmer. We really should care about it as a community, as a society, to make sure that there is a way that we can produce a lot of that protein very inexpensive and with a low impact on the environment.